Welcome to the Deep Dive. Today, we're venturing into an absolutely incredible world, the microscopic universe within a single human cell. It's the fundamental unit of life, and understanding it is key to, well, understanding everything about us.
It really is. The complexity is just astounding.
So our mission today is to take a deep dive using a fantastic resource, Histology, a Texan Atlas, 8th edition. We'll break down the cell's parts, figure out what they do, and even look at some really interesting clinical connections. Think of it as your shortcut to the inside story.
And it's so crucial. Understanding the cell helps us grasp everything from how we fight infections to how we grow. And, what's amazing is how diverse cells are. Skin cells, nerve cells, muscle cells, yet they all share these core life mechanisms.
Absolutely. It promises to be full of those aha moments.
Wow.
So let's jump right in. Okay, let's unpack this. We say cell, but what's actually in there? What are we looking at?
Broadly, you've got two main areas. The nucleus, which holds the genetic material, and then everything else, which we call the cytoplasm.
And we're focusing mostly on the cytoplasm today.
Exactly. It's this busy, watery space outside the nucleus. It's packed with tiny structures called organelles, like miniature organs, plus a whole internal scaffolding system, the cytoskeleton, and various bits and pieces called inclusions, all floating in this gel-like matrix.
Organelles, like little organs. I like that.
Yeah, and specific functions are really tied to these structures. Think about muscle cells. They're specialized for contraction, right? So inside, they're loaded with specific proteins organized perfectly for that job.
So the structure totally dictates the function.
Precisely. You can often tell a cell's job just by its shape or what's inside it. Kidney tubule cells look different from big nerve cells, which look different from smooth muscle cells. Form follows function, right down to this level.
And these organelles, these tiny organs, they come in different types.
Right. Some are wrapped in their own membranes. We call them membranous organelles. This creates compartments separating different chemical environments. It's super efficient, increases surface area for reactions, and keeps potent things like digestive enzymes and lysosomes safely contained. Containment. Makes sense.
Then you have the non-membranous ones, no membrane wrapping around them. Think ribosomes, parts of the cytoskeleton.
Got it. Okay, let's zoom right out to the edge. The plasma membrane. It's not just a simple container, is it?
Oh, definitely not. It's incredibly dynamic. Think of it as a fluid double layer of lipids fats, essentially with proteins embedded in it or attached to its surface. It's what we call the fluid mosaic model.
Fluid mosaic, meaning things can move around within it.
Exactly. The lipids provide the basic structure, but they're quite mobile, and the proteins can shift around too, though sometimes they're anchored. And these proteins do a huge range of jobs.
And there's that fuzzy coat on the outside too, right? The glycoalexis?
Yes, that's made of carbohydrate chains attached to the membrane proteins and lipids. It's like the cell's ID badge and communication antenna combined, crucial for cell recognition, receiving hormone signals, all sorts of interactions.
So it's how cells talk to each other, in part.
In large part, yes. And within the membrane itself, there are these specialized zones called lipid rafts. Lipid rafts, like little floating platforms.
Kind of. They're slightly thicker, less fluid areas rich in cholesterol and specific lipids. They act as organizing centers, bringing together receptors and enzymes needed for quick signaling, like concentrating all the key players in one spot.
Efficient.
Very. But clinically, they can be a vulnerability. Some bacteria, like Shigella or Salmonella, and even viruses, have figured out how to hijack these rafts to get inside cells or evade the immune system.
Wow. So our own cell structures can be turned against us.
It happens. It highlights how complex the host-pathogen interaction is.
And the proteins embedded in the membrane. What are their main roles? You said they do a lot.
Oh, huge variety. Some are pumps, actively moving ions or molecules against their concentration gradient using energy. Think of the sodium-potassium pump. Vital for nerve cells.
Okay, pumps. What else?
Channels. These create pores that allow specific ions or water to flow through passively down their gradient. Some are always open, others are gated, opening or closing in response to signals.
Like tiny gates.
Exactly. Then you have receptors. They bind to specific signaling molecules, like hormones, on the outside and trigger a response inside the cell.
Communication central.
Absolutely. And linkers these proteins anchor the cell's internal cytoskeleton to the outside environment or to other cells, think integrins.
Holding everything together.
Right. Some membrane proteins are enzymes carrying out chemical reactions right there at the membrane surface, like ATPase is involved in energy use or digestive enzymes on gut cells. And finally, some are purely structural, just contributing to the membrane's integrity. It's a busy place.
Incredibly busy. So If the membrane is this dynamic gatekeeper, controlling what comes in and interacts,
Hmm.
how does the actual transport happen? How do things cross this barrier?
Well, it depends on the substance. First, there's communication cell signaling. An external signal binds a receptor, triggering a cascade of events inside signal transduction, often involving modifying proteins to amplify the message and cause a specific response.
Like a relay race inside the cell.
A very fast, complex relay race, yes. For actual movement across the membrane, small, uncharged things like oxygen or CO2 can just diffuse through the lipid layer. Simple diffusion.
But most things can't do that.
No. Most need help from transport proteins. You have carrier proteins that bind a specific molecule, change shape, and shuttle it across. This can be passive or active, requiring energy.
Okay, carriers.
And channel proteins, which form those hydrophilic pores we mentioned. They're often highly selective for specific ions like sodium or potassium channels, and their opening and closing is tightly regulated.
Got it. But what about bigger stuff like large proteins or even bacteria that can't fit through a channel?
Right. For large cargo, the cell uses vesicular transport. It's about forming membrane-bound sacs or vesicles. Bringing things in this way is called endocytosis.
Endocytosis, taking things inside.
Yep. And releasing things out is exocytosis. These two processes are actually coupled. The cell needs to balance the membrane being added during exocytosis with the membrane being removed during endocytosis. If one stops, the other usually does too.
A balancing act for the membrane budget.
Exactly. Now, endocytosis isn't just one thing. There's penocytosis, cell drinking. The cell gulps in small amounts of extracellular fluid and dissolved molecules non-specifically.
Just sampling the environment?
Kind of. Then there's phagocytosis, cell eating. This is for big particles like bacteria or cellular debris. The cell extends projections called pseudopods to engulf the target, forming a large vesicle called a phagosome. Immune cells are pros at this.
Eating invaders. Makes sense.
And finally, receptor-mediated endocytosis. This is highly specific. Molecules bind to specific receptors on the cell surface, which cluster together in specialized regions called clathrin-coated pits. These pits then invaginate and pinch off to form vesicles, bringing in only the targeted cargo. It's very efficient for importing specific things the cell needs, like cholesterol via LDL receptors.
Super selective intake. Okay, so that's getting stuff in. What about getting stuff out? Exocytosis.
Right, the exit strategy. Two main routes here. The constitutive pathway is basically continuous secretion. Things are packaged and sent out as soon as they're ready. Think of plasma cells constantly releasing antibodies. No storage involved.
Just a steady stream out.
Then there's the regulated secretory pathway. This is used by specialized cells like hormone-producing cells or neurons. They package their products, hormones, neurotransmitters, into secretory vesicles and store them. Release only happens when the cell receives a specific signal, often involving an influx of calcium ions.
So release on demand.
Precisely. And the targeting of these vesicles, both for endo and exocytosis, is incredibly precise. It involves specific protein markers on the vesicle, V snares, and the target membrane, T snares.
The snares you mentioned earlier.
Yes. They act like molecular address labels and docking clamps. They ensure the vesicle fuses only with the correct destination membrane. It's a beautiful, intricate system.
And the one targeted by those toxins.
Yeah, exactly. Botulinum toxin, Botox, and tetanus toxin are potent neurotoxins because they specifically cleave snare proteins at neuromuscular junctions. Botox blocks acetylcholine release, causing flaccid paralysis, which is why it's used therapeutically in small doses. Tetanus toxin blocks the release of inhibitory neurotransmitters, leading to uncontrolled muscle contraction, the spasms of tetanus. It really underscores how vital this trafficking system is.
Wow. Tiny proteins, huge consequences if they're disrupted. So, okay, materials come in via endocytosis. Where do they go next? Do they just float around?
No, they enter the endosomal pathway. They first arrive in early endosomes, which are sorting stations usually near the cell periphery. Here, decisions are made. Are the receptors recycled back to the plasma membrane? Is the cargo sent on for destruction?
Sort in the mail.
Pretty much. Some material moves on to late endosomes, which are typically closer to the nucleus and more acidic. These often mature into or fuse with lysosomes.
The recycling centers or maybe the incinerators.
More like the digestive system. Lysosomes are sacs filled with powerful hydrolytic enzymes, proteases, lipases, nucleases that can break down virtually any biological macromolecule. They maintain a very acidic internal pH, around 4.7, which is optimal for these enzymes.
How does the lysosome itself not get digested?
Great question. Its membrane is special. It contains unique lipids and heavily glycosylated proteins, like LMPs, that protect it from the acidic environment and the enzymes inside.
Clever design.
Absolutely essential. And this system is critical for health. When it breaks down, you get lysosomal storage diseases, or LSDs.
LSDs. These are genetic.
Yes, usually inherited genetic defects where a specific lysosomal enzyme is missing or doesn't work properly. As a result, the substance that enzyme was supposed to break down accumulates inside the lysosome, eventually clogging up the cell and causing damage.
Like Tay-Sachs disease.
Tay-Sachs is a classic tragic example. An enzyme needed to break down a specific lipid, GM2 ganglioside, is missing. This lipid builds up, especially in nerve cells, leading to progressive neurodegeneration and early death. There are many different LSDs affecting different enzymes and causing a wide range of symptoms, but the underlying principle is lysosomal dysfunction.
That really highlights their importance. It also explains why therapies like enzyme replacement are being developed.
Exactly, trying to supply the missing enzyme.
Okay, so lysosomes handle external waste and ingested material. What about the cell's own internal junk? Old organelles, damaged proteins.
That's handled primarily by autophagy, which literally means self-eating. It's a crucial process for clearing out damaged components and recycling their building blocks, especially during stress or starvation.
How does that work? Does the cell just eat itself?
In a controlled way. The most common type, macroautophagy, involves a membrane forming around the chunk of cytoplasm or organelle destined for removal, creating a double membrane vesicle called an autophagosome. This then fuses with a lysosome and the contents are digested.
So it wraps up its own trash and delivers it to the lysosome.
Essentially, yes. There are other more selective types of autophagy too, but it's a fundamental quality control and survival mechanism.
And is there another way besides lysosomes to get rid of unwanted proteins?
Yes, there is. For many individual proteins, especially those that are misfolded or damaged or just no longer needed, the cell uses proteasome-mediated degradation.
Proteasomes, what are they?
They're large barrel-shaped protein complexes found in the cytoplasm and nucleus. They act like molecular shredders. Proteins targeted for destruction get tagged with a chain of small proteins called ubiquitin.
Ubiquitin, like a tag for disposal.
Exactly. The polyubiquitin chain acts as a signal. The proteasome recognizes this tag, unfolds the protein, and chops it into small peptides using ATP for energy.
So two main waste disposal systems. Lysosomes for bulk cargo and autophagy, and proteasomes for specific tagged proteins.
That's a good summary. And again, clinical relevance. Problems with the ubiquitin proteasome system are linked to diseases like Alzheimer's, where protein aggregates build up, and Parkinson's. On the flip side, some cancer treatments actually inhibit the proteasome because cancer cells often rely heavily on it to degrade proteins that would otherwise trigger cell death. Burazomin for multiple myeloma is a key example.
Fascinating how understanding these basic processes leads directly to therapies. Okay, let's shift gears. We've talked transport and breakdown. What about building things? Where does protein synthesis happen, and how does the cell make energy, the factories and power plants?
Right. Let's start with the factories. Protein synthesis begins with the rough endoplasmic reticulum, the RER. It's called rough because its surface is studded with ribosomes, which are the actual protein-making machines. Structurally, it's a network of interconnected membrane sacs called cisternae, often continuous with the outer membrane of the nucleus.
So the ribosomes make the proteins, and they go into the RER.
Proteins destined for secretion or for insertion into membranes or delivery to certain organelles like lysosomes usually start their journey here. The mRNA recipe from the nucleus is read by a ribosome. If the protein being made has a specific signal peptide, like an address label, the ribosome gets directed to the RER membrane. The growing protein chain is then threaded into the RER lumen or inserted into its membrane.
And once inside the RER.
It's not just dumped there. Inside the RER, proteins start to fold into their correct three-dimensional shapes, often aided by chaperone proteins. Disulfide bonds might form, and initial sugar chains, glycosylation, can be added. It's also a critical quality control point.
Quality control how?
If a protein misfolds, the RER tries to fix it. If it can't be fixed, it's actually ejected back into the cytoplasm, tagged with ubiquitin, and sent to the proteasome for destruction. It's called ER-associated degradation, or ER.
So the cell has mechanisms to prevent faulty products from getting out.
Yes, crucial ones. But sometimes they fail, or the problem's overwhelming. In alpha-1 antitrypsin deficiency, for instance, a genetic mutation causes the A1AT protein to misfold and accumulate within the RER of liver cells, damaging them. The lack of functional A1AT reaching the lungs also leads to damage there, causing emphysema.
Another direct link between cellular machinery and disease.
Absolutely. Now, adjacent to the RER, you often find the smooth endoplasmic reticulum, or SER. No ribosomes here, hence smooth.
Different factory floors.
Different functions entirely. The SER is big in cells that synthesize lipids, like steroid hormones, think adrenal cortex cells or Leydig cells in the testes. It's also the primary site for detoxification in the liver, packed with enzymes like cytochrome P450 that modify and neutralize drugs and toxins, and it stores calcium ions.
Detox and lipid production. Okay, where do things go after the ER?
To the Golgi apparatus, or Golgi complex. Think of it as the cell's finishing and shipping department. It's a stack of flattened, membrane-bound sacs called cisternae.
Like a stack of pancakes.
A bit, yes. Proteins and lipids arrive from the ER at one face of the Golgi, the cis face, travel through the stack undergoing further modifications. Sugars might be trimmed or added, proteins might be cleaved, and then they're sorted and packaged into vesicles at the other face, the trans face, for delivery to their final destination.
So refining and addressing the packages.
Exactly. The Golgi directs proteins to the lysosomes, or to different parts of the plasma membrane, or packages them into secretory vesicles for release outside the cell. It's central to the secretory pathway.
Okay, factories covered. What about the power plants? Mitochondria?
Ah, yes, the mighty mitochondria. These are the primary sites of ATP production through aerobic respiration. They generate the vast majority of the cell's energy currency.
So they're essential for almost everything a cell does.
Absolutely. Cells with high energy demands, like muscle cells or neurons, are packed with mitochondria. They're fascinating organelles, often sausage-shaped, and quite mobile within the cell.
And they have that interesting evolutionary story, right?
They do. The endosymbiotic theory proposes that mitochondria evolved from free-living aerobic bacteria that were engulfed by an ancestral eukaryotic cell billions of years ago. Evidence for this includes the fact that mitochondria have two membranes, they have their own circular DNA, similar to bacterial DNA, and their own ribosomes and protein synthesis machinery.
Like a cell within a cell.
Almost, although most mitochondrial proteins are actually encoded by the cell's nuclear DNA and imported. Structurally, you have the smooth outer membrane and then the inner membrane, which is highly folded into shelf-like projections called cristae.
Why the folds?
To dramatically increase the surface area. Because embedded within that inner membrane are the protein complexes of the electron transport chain and the ATP synthase enzyme, the machinery that actually makes ATP.
That's where the energy generation happens.
Precisely. The space inside the inner membrane is the matrix, containing enzymes for other metabolic pathways like the citric acid cycle, plus the mitochondrial DNA and ribosomes. The electron transport chain pumps protons from the matrix into the space between the two membranes, creating a proton gradient. The flow of protons back into the matrix through ATP synthase drives ATP synthesis. It's called chemiosmotic coupling.
A very efficient energy conversion process.
Incredibly efficient. But like any complex machine, it can break down. Mitochondrial diseases often arise from mutations in either mitochondrial DNA or nuclear DNA encoding mitochondrial proteins. These typically affect tissues with high energy needs.
Like the MRF condition you mentioned.
Yes. Myoclonic epilepsy with ragged red fibers, MRF, is a classic example caused by a mutation in a mitochondrial tRNA gene. It leads to muscle weakness, seizures, ataxia problems in high-energy tissues. The ragged red fibers seen in muscle biopsies are actually clumps of abnormal mitochondria. Mitochondria are also involved in triggering apoptosis, programmed cell death, by releasing factors like cytochrome C when the cell is stressed or damaged.
So they're central to both life and death decisions for the cell.
You could say that. Finally, briefly, we should mention peroxisomes.
The often forgotten one.
Sometimes overlooked, yes, but vital. These are small, single-membrane organelles containing oxidative enzymes, most notably catalase. Their key job is to break down fatty acids and, crucially, to neutralize hydrogen peroxide, H2O2, a toxic byproduct of metabolism.
So they handle specific toxic waste.
Exactly. They detoxify H2O2 into water and oxygen. They also play a role in lipid synthesis and alcohol detoxification in the liver. And again, defects are serious. Zellweger syndrome is a devastating disorder where peroxisomes fail to form or function correctly, leading to severe neurological problems and early death.
It really drives home how interconnected and essential all these tiny parts are. Okay, we've covered organelles. What about the cell's internal structure? The scaffolding.
Right, the cytoskeleton. It's not just a bag of organelles. Cells have an intricate internal framework that provides shape, support, organization, and enables movement. Think of it like the cell's bones and muscles combined.
Bones and muscles. So it's dynamic.
Very much so. It's made of three main types of protein filaments. Let's start with the largest, microtubules.
Microtubules, what are they like?
They're relatively rigid, hollow tubes made of polymerized tubulin protein subunits. They act like structural girders, helping maintain cell shape, but they're also incredibly dynamic. They can grow and shrink rapidly by adding or removing tubulin subunits, a process called dynamic instability.
Growing and shrinking, like little construction beams being adjusted.
Exactly. This allows the cell to quickly reorganize its internal structure. Microtubules typically radiate out from a central point called the microtubule organizing center, or MTOC, near the nucleus.
And what do?
Lots. They form the tracks along which motor proteins like kinesins and dyneins transport organelles and vesicles around the cell like a cellular railway system. They're the core structure of cilia and flagella, enabling movement. And crucially, they form the mitotic spindle that separates chromosomes during cell division.
The railway tracks and the chromosome movers. Got it.
And defects have consequences. Cardiganer syndrome, for example, involves defects in dynein arms, in cilia and flagella, leading to respiratory problems and infertility. Also, many cancer drugs like vincristine or paclitaxel, Taxol, target microtubule dynamics to stop cancer cells from dividing.
So they're a key therapeutic target. What's the next filament type?
The thinnest ones, actin filaments, also called microfilaments. These are flexible, solid rods made of polymerized actin protein. They're often concentrated just beneath the plasma membrane, forming a supportive meshwork called the cell cortex.
Thinner and more flexible than microtubules.
Yes, and like microtubules, they're dynamic and polar, with actin subunits adding at the one end, the plus end, and often dissociating from the other, the minus end, a process called treadmilling. Their assembly and organization are controlled by a whole host of actin-binding proteins.
What are actin filaments involved in?
So many things. They provide mechanical support, especially at the cell surface. They form the core of microvilli, those finger-like projections that increase surface area. They're essential for cell movement. The crawling of cells involves actin polymerization, pushing the leading edge forward. And they're involved in muscle contraction, working with myosin motor proteins.
Cell crawling and muscle power.
Exactly. And clinically, some pathogens exploit this. The bacterium Listeria monocytogenes actually hijacks the cell's actin polymerization machinery to build an actin comet tail that propels it through the cytoplasm and even into adjacent cells.
Wow, using the cell's own engine against it.
It's a remarkable example of pathogenic adaptation. The third type is intermediate filaments.
Intermediate, so size-wise between the other two.
Exactly, around 8–10 nanometers in diameter. Unlike microtubules and actin, these are generally much more stable and less dynamic. Think of them as the cell's ropes or cables. Their main role is providing tensile strength and resistance to mechanical stress.
The stress-bearing component.
Precisely. They're particularly important in tissues subjected to physical forces, like skin epithelium or muscle. There are different classes based on the proteins they're made of. Keratins are found in epithelial cells, skin, hair, nails. Vimentin is common in mesenchymal cells. Neurofilaments provide structural support in nerve axons, and nuclear lamins form a supportive meshwork inside the nuclear envelope.
So different ropes for different jobs and different cell types.
You got it. And they often connect to cell junctions like desmosomes and hemidmosomes, anchoring cells to each other and to the extracellular matrix, creating strong tissues.
And problems here also cause disease.
Yes. For instance, mutations in keratin genes can cause blistering skin disorders like epidermolysis bullosa simplex, where the skin is extremely fragile. Abnormalities in neurofilaments are implicated in neurodegenerative diseases like ALS and contribute to the neurofibrillary tangles in Alzheimer's disease. Accumulations of intermediate filaments like the GFAP protein in Alexander disease or the keratin-rich Mallory bodies in alcoholic liver cirrhosis are often diagnostic markers of specific cell stress or injury.
It really shows how this internal architecture is vital for function and health. You mentioned the MTOC organizing microtubules. What about centrioles?
Right. Centrioles are typically found within the MTOC in animal cells. They're small, paired, barrel-shaped structures made of triplet microtubules. The MTOC itself, containing the centrioles and surrounding pericentriolar material, is the main site where microtubules are nucleated or born. It controls their number and orientation.
What's the specific job of the centrioles themselves?
They have two key roles. First, they form basal bodies, which are essential templates for the assembly of cilia and flagella. Second, during cell division, the MTOC, with its centrioles, duplicates and moves to opposite poles of the cell, organizing the formation of the mitotic spindle that pulls the chromosomes apart.
So critical for movement and division.
Absolutely. And errors in central duplication are a big deal. Having too many centrioles can lead to abnormal multipolar spindles during mitosis. This often results in incorrect chromosome segregation, aneuploidy, which can kill the cell, but sometimes contributes to the genomic instability seen in cancer. Extracentrosomes are actually a common feature of many human tumors.
Another link to cancer. It seems like so many basic cellular processes, if they go wrong, can contribute to cancer.
That's very true. Cancer is often a disease of disrupted fundamental cell biology, uncontrolled division, faulty repair, abnormal signaling, resistance to death signals.
It's incredible how these tiny filaments and organizing centers create such complex architecture and enable such vital functions.
It really is. And thinking about how these dynamic microtubules, flexible actin filaments, and stable intermediate filaments all interact and cooperate to give each cell its unique properties, it's a fascinating area of ongoing research. How does this intricate network respond to signals and maintain tissue integrity? And what really happens at the molecular level when it breaks down?
So much still to learn. Okay, just to round things off, besides organelles in the cytoskeleton, what else is floating around in the cytoplasm? You mentioned inclusions.
Right, inclusions. These are basically non-living accumulations of material within the cytoplasm, not enclosed by a membrane in the same way organelles are. They're often stored nutrients or metabolic byproducts.
Like what?
Common examples include glycogen granules, which are stored glucose, especially prominent in liver and muscle cells. Lipid droplets, stored fats for energy, are common in fat cells, but also found in others. Then there's pigments like lipofuscin, a brownish wear and tear pigment that accumulates with age, representing residues of lysosomal digestion. Or hemosiderin, an iron storage complex often seen after bleeding. Sometimes you even see crystalline inclusions, though their function isn't always clear.
So stored food, waste products, pigments, miscellaneous stuff.
Pretty much. And all of this organelles, cytoskeleton inclusions, is suspended in the cytoplasmic matrix, or cytosol.
The background jelly.
Essentially, yes. It's the concentrated aqueous solution that fills the cell. It's where many fundamental metabolic pathways occur, like glycolysis, and where ribosomes synthesize proteins destined to stay in the cytosol itself. It's not just passive goo, though. It's highly organized and crowded with molecules.
What a tour. From the outer membrane, through transport systems, digestive centers, factories, power plants, scaffolding, and storage.
Yeah.
It's truly mind-boggling what goes on inside a single cell.
It really is a microscopic city, bustling with activity, highly organized, and incredibly efficient. And understanding this city plan, how all the parts work together, is absolutely fundamental to understanding physiology and pathology.
It makes you realize that health and disease often boil down to what's happening or going wrong at this incredibly small scale.
Exactly. A tiny defect in one protein, one pathway, can have massive consequences for the whole organism. It underscores the elegance and also the fragility of the system.
Well, hopefully this deep dive has illuminated some of that elegance for you, our listeners. When you think about the cell now, what aspect of its design or function really stands out to you?
And it's a field that's constantly evolving. We're learning more every day about how these components interact, how they're regulated, and how we might intervene when things go wrong. The secrets to new therapies are often hidden within these cellular details.
Thank you so much for taking this journey with us into the cell. And thank you, as always, for being part of the Last Minute Lecture family.